Welcome to Chapter 4, Biocatalysis. This chapter is about enzymes. This chapter consists of three subtopics: 4.1, Properties of Enzyme and Mechanism of Actions, 4.2, Cofactor, and 4.3, Inhibitor. Did you know that enzyme can help to fight disease? Fungi, bacteria, parasite, and even a virus are made up of protein. Although enzymes are themselves protein, there is enzyme that can break down other protein, which is known as protease. High level of protease in the blood can help to eliminate all the disease-causing vector in our body. So, enzymes are not only for digestion. Isn't that amazing? 4.1, Properties of Enzyme and Mechanism of Action. At the end of this subtopic, you should be able to state the properties of enzyme, state the 6 classes of enzyme according to International Union of Biochemistry and Molecular Biology classification, explain how enzyme lowering activations energy, illustrate to explain the mechanism of enzyme action based on induced feed model, state the factor that affect the enzymatic reaction. A catalyst is a substance that speeds up a chemical reaction without being affected chemically in the reaction. Enzymes are biological or organic catalysts which speed up a reaction without undergoing any permanent change themselves. Properties of enzyme includes: enzyme is highly specific. Enzyme is not altered during reaction. Only small amount of enzyme is required to catalyze a reaction. Enzyme is not damaged or destroyed by reaction it catalyzes. Enzyme has no effect on the thermodynamic of the reaction. Enzyme is denatured by high temperature and high pH. Enzymes speed up the chemical reaction. Enzymes are normally complex three-dimensional globular protein. Therefore, as a protein, obviously the characteristics of enzyme reflect the properties of proteins. Then it means that it can be dissolved in water and can be diluted with the use of solution. Enzymes are classified according to IUBMB, the International Union of Biochemistry and Molecular Biology, and is based on the reaction they catalyze. Enzymes are usually named by adding the suffix ase, example, amylase, protease, ligase, etc. The 6 categories of enzymes are hydrolase, oxidoreductase, lyase, isomerase, transferase, and ligase. Oxidoreductase catalyzes oxidation-reduction reaction by transferring hydrogen or oxygen atoms or electron from one molecule to another. Example of these enzymes is dehydrogenase, which catalyzes the reaction by transferring hydrogen from one substrate to another hydrogen acceptor. Another example is oxidase, which catalyzes oxidation in the presence of free oxygen. Transferase are enzymes that catalyze the transfer of specific group like methyl, acyls, amino from one molecule to another. The example of this enzyme is transaminase, which transfer amino group, and phosphorylase, which add inorganic phosphate. Hydrolase catalyzes hydrolysis reaction in the presence of water. This is the most common class of enzyme. Hydrolase split chemical bond like peptide bond, glycosidic bond, and ester bond by hydrolysis. Example, hydrolysis of a disaccharide into 2 monosaccharide. Ligase are involved in eliminations or addition reaction in which a group of atoms is removed or added to a substrate. Additions or removal of a functional group is without additions of water. Example of ligase is pyruvate decarboxylase. Carbon dioxide is removed from pyruvate to form ethanol. Isomerase catalyzes molecular isomerization. They catalyze the internal rearrangements of atoms in a substrate and convert one isomer to another. Example of enzyme in this class is phosphoglucomutase. Phosphoglucomutase convert glucose 1-phosphate to glucose 6-phosphate. Ligase catalyze formations of bond between two molecules. Ligases are also known as synthetase. They are involved in the formations of covalent bond. ATP is needed to form this bond. Example of ligase is aminoacyl tRNA synthetase. This enzyme involved in the formations of aminoacyl tRNA in protein synthesis. Let's share and remember the 6 classes of enzyme: hot yields, hydrolase, oxidoreductase, transferase, isomerase, lyase, ligase. The energy required for substance to react is called activation energy. Activation energy is the minimum energy which is needed to break or make the bond in a molecule and start a reaction so that an effective reaction can occur. It can be provided in the form of heat that substrate absorbs from the surrounding, but high temperature can kill cells. Enzymes speed up metabolic reaction by lowering energy barrier. Enzymes lower activation energy so that the transition state can be reached at cellular temperature. Without an enzyme, a high temperature is required. Transition state is the unstable conditions of reactant molecule that have absorbed sufficient free energy to react. Reactant must absorb enough energy to reach the transition state. The enzyme acts by forming an unstable enzyme-substrate complex with the substrate. This complex is subsequently transformed into the product with the release of the enzyme. This figure showing a higher activation energy for an uncatalyzed reaction than for a catalyzed reaction. Enzymes do not change the nature of the reaction, but only speed up the reaction. Note that change in free energy remain the same for both types of reaction. Enzymes are normally larger than the substrate molecule that act open. The part of the enzyme molecule that come into contact with the substrate is called the active site. An enzyme specificity is caused by the shape of the active site. An enzyme combines with its substrate to form an enzyme-substrate complex, which then dissociate into product and enzyme. The chemical reaction within living organisms are regulated by controlling the point at which catalysis takes place. The unique three-dimensional shape of an enzyme enable it to stabilize a temporary association between substrates. By bringing two substrates together in correct orientations or by stressing particular chemical bond of a substrate, an enzyme lowers the activation energy. The reaction thus proceeds much more quickly than it would without the enzyme. The active site of the enzyme fits the shape of its substrates. When the substrates slide into the groove of the active sites, the protein is induced to alter its shape slightly and bind the substrate more tightly. These alterations of the shape of the enzyme to better fit the substrate is called induced fit. The enzyme itself is not changed or consumed in the reaction. So, when products are formed, enzyme is free to bind other substrates. Did you know aging will decrease the productions of enzyme? Our body will produce less protease, lipase, and amylase as we get older, which means digestion of protein, fats, and carbohydrate can be impaired.